Good morning.
Good morning, Nitika.
Good morning, Daniel. I'm so sorry for the delay. I had to change the browser. It wasn't
Ah,
working on Chrome and it started working on Firefox.
good. Well, we're here. We're happy to have you here. With that being said, let's go ahead and hand over the microphone. I want to introduce Dr. Palmer. She is our program director for our Masters in Biotechnology and Stem Cell program, and she is the host, and she will be showcasing the great work of our students in our master's program. I hope you guys enjoy the presentation today. They did a great job. I got to see them yesterday.
Wonderful. Well, thanks so much. Not sure why my background was so dark, but anyway, I'll be turning off my video and sharing the slideshow. So, Daniel, I am the co-host, correct?
You're the host.
Okay, great, great. Well, thank you so much, everyone, for coming. And my apologies, sincere apologies for the delay. There was some issue with the Zoom playing on one platform and not on the other. And my name is Nitika Parmar, and I'm the current program director of the MS Biotechnology program at CI, as well as the director of the CERM stem cell program. And this presentation is going to be talking about the power of stem cells. There's a lot that can be discussed here, and I will make a presentation first for about 45 to 50 minutes, followed by three groups of our graduate students who will be talking about the utilization of cells for potential use for fixing different kinds of diseases. So please bear with us as we load our presentation. And I'm going to turn off my video and share my screen at this point. Please let me know when you can see this. Is everyone able to see this? Can I just need a yes? Can everyone see the PowerPoint?
Yes.
Okay, great, great. Thank you so much for confirming. All right. So, as I mentioned here, I am in the biology program, and our talk today is about stem cells and the power of stem cells. So, if you look back at the covers of the Time magazine for the past 20 or 25 years, since the inception of the cloning, which happened almost 50 years ago, there's been a lot of hype around the process known as cloning, and we'll talk about that in just a few minutes. Cloning humans, cloning animals, producing twins, what makes each one of us special, how are we distinct from each other, how are our genes connected, how are we different. So there's a lot going on in the area of genomics. And genomics simply refers to the study of your genes, basically the study of the DNA, which is residing in our cells. And it's very interesting because 99.5% of our DNA in the human population is identical, regardless of our geographical location, our race, our skin color, our different kinds of upbringing. 99.5% is identical. But it's that 0.5%, which is very different amongst all of us, that makes us quite unique in the way we behave and the way we look. So the whole question is stem cells, to use or not to use. Are they a blessing in disguise or are they evil? And that's what we are going to discuss in this particular seminar here. So I'll start with very basic concepts just to give some of you an idea what the term stem means, since the stem cell world is very loosely used quite often by the media today. So the word stem simply means a fundamental line from which others have arisen. So we can talk about something which is the original from which others have been arising or reproducing. So in scientific language, we call the stem cell as a progenitor cell from which all the other progeny are arising. So it's the most unique and the most original cell in our body. So how is a stem cell different from our regular cells in the body? And how many cells do we have in the body in the first place? You'll be surprised to know, on an average, a human body has roughly close to 3 trillion cells, right from the top of our head all the way to our heels. We are abounding in 3 trillion cells, of which some of them are stem cells. A very small population are stem cells. The majority are differentiated cells. And when I use the word differentiated, I am referring to how these cells are acquiring different kinds of functions. Some of the cells are meant to give us vision. Some of the cells in the stomach are meant for digestion. Some of the cells in the pancreas are
Sorry,
meant
Dr.
for
Palmer.
secretion of insulin and so forth. So we have a huge population of very specialized cells in our body. And this
Hello, Dr. Parmar.
specialization,
Excuse me.
Excuse me, Dr. Parmar. Sorry to interrupt you.
please mute
Uh,
yourself, all the students.
The slides are not moving. We only see your first slide.
Can you see it now?
No,
It's your
we
first
can't.
slide.
Can you see this slide which says what is a stem cell?
No, we only see your first slide. It's not changing.
Okay, let me read it. That's a bit strange.
Now we see it. Yep.
Okay, let me start sharing all over again. Thank you for letting me know. Can you see this?
Yes.
Yes. Yes.
Okay, so I think something happened when I went in the presentation mode for some reason, but I will probably play it in the non-presentation mode as long as you can see it. So thanks for letting me know. So what exactly is a stem cell and how is it different from a differentiated cells? Now remember, differentiated cells are very specialized cells. They're very unique. They're present only in certain areas of a body. For instance, the stomach cell will only be present in the stomach. It's not going to be present in the bones or the skin. Likewise, the skin cell is going to be only on the surface of your body, not in the pancreas. So these are very specialized cells. Now, the stem cell is quite distinct from differentiated cells. One, the stem cell has a capacity, what we call self-renewal. That means it has an unlimited capacity to keep on dividing and produce more copies of itself. So on the left side, you can see I've used the word self-renewal, which means it can reproduce itself unlimitedly because it has that capacity. Although it does not do so because it follows the second pathway where certain signals in the body tell the stem cell, okay, now it's time for you to become more functional. And that process is known as differentiation. So there's various proteins which act as messengers, as signals, which act on the stem cell and tell it to divide and differentiate, not simply divide, but to divide and differentiate. So there's two pathways, one where it makes copies of itself, and two, where it actually makes a copy and then differentiates. And that is known as specialization. So stem cells are extremely valuable for us in the body because these are the only cells in the body which can practically turn into any cell type that you want them to be turning into. They can become a muscle, they can become a kidney cell, they can become a gallbladder cell, they can become a neuron, they can become anything they want to, but depending on the signals that are being provided to them. So that process is known as differentiation. So there are two very specific properties of stem cells. The first one I already mentioned, the process known as self-renewal. So stem cells have a capacity to basically reproduce themselves for a very, very long period of time, almost immortalized, almost, I would say. But in addition to that, they have what we call unlimited potency. That means they can be induced when certain protein signals are given to them under very controlled conditions to become cells with very specialized functions. So these two features, self-renewal and unlimited potency, are exclusively given or designated to stem cells, not any other cells in the body. Other cells can either self-renew and not have unlimited potency, And some other cells can have unlimited potency, but not self-renew. But stem cells are the only cell which have both of these features. And that's why they're so valuable in our body. And that's why we use them in our science too. So why do they self-renew and differentiate? Well, self-renewal is very important to maintain a pool of stem cells which are hiding in our bone marrow and other very deep crevices in the tissues, so that whenever there is an injury in our body and we are required to produce fast new cells in a very fast manner, the stem cells kick in and try to replenish the damaged and the lost cells. So that is very important for regular tissue repair in our body. And the other most important reason is if the stem cells do not differentiate, then we will not be specialized and we will not be able to perform our bodily functions. So both capacities, self-renewal and differentiation, are very, very important for stem cells to function. Now I'm going to talk about the word potency. You must have heard of the word stem cells are very potent, they are pluripotent, they are totipotent, they are multipotent. So what does the word potency mean? So with regard to stem cells, we use three different kinds of terminologies. One, pluripotent; second, multipotent; and third, unipotent. So the word pluri means different kinds of cell types, unlimited cell types. Multi means several, but not unlimited. And uni means single. So a pluripotent stem cell is one which can specialize into any cell type in the body. A multipotent stem cell is one which can produce different kinds of cells, but all those cells fall in one family of cells. They cannot like turn into any kind of cell, but they can form different groups of cells. And I'm just going to explain this a bit more. And unipotent means they can only produce one cell type. So the classic example of a pluripotent stem cell is the embryonic stem cell, which, as the name suggests, comes from the human embryos. And these have a capacity to differentiate into any cell type, and rightfully so, because once the embryo grows in the fetus, it has to specialize quickly to form fingers, to form brain, to form stomach, to form feet. So those embryonic stem cells have a capacity to turn into any cell type in the body, and those are the most valuable. The multipotent can produce different kinds of cells, but only belonging to one family. And the classic example is the cells in our blood system. So the blood stem cells are known as multipotent because they can specialize into red blood cells, they can specialize into white blood cells, and they can specialize into platelets, which are involved in a clotting mechanism. So within a family, those are known as multipotent. And the unipotent, as the name suggests, is this is a very unique stem cell only found in very specific tissues, which can only produce cells of that tissue and nothing else. So these are sort of what we term as single lineage. They produce only one kind of cell from the original stem cell. So what are the various types of stem cells? Initially, we talked about the different potencies of stem cells and what are the various general category of stem cells. So broadly, they fall into four different categories. The first one is embryonic stem cell, commonly referred to as ESCs. The second is adult stem cells. The third is cord blood stem cells. And the fourth is amniotic fluid derived stem cells. So let's look at each one of them. So as the name suggests, the embryonic stem cells are coming from the embryo. So what is an embryo? An embryo is the outcome of the fusion of the male sperm with the female egg in the body, which actually attaches to the uterus. Initially, it's just a one cell, which is known as a zygote, which actually starts dividing and turns into an early blastocyst and then turns into a late blastocyst. And if you see this figure, the orange cells in the center of this ball of cells are the most valuable cells in the human body. This is known as the inner cell mass. And these are the cells which are known as the embryonic stem cells. These orange cells have the capacity to divide unlimitedly into different kinds of cells in the human body. And this ball is formed pretty much at the fourth or the fifth day of the embryo post-fertilization. So it's an extremely early stage of the embryo. And that's why there are ethical concerns in using embryonic stem cells, because in order to get these orange cells out of this blue ball, one is required to destroy the embryo. That means end the human life. And that is why the whole ethical concern comes into play here. And we'll discuss the ethics maybe in about 15 minutes time here. So this is a very, very cool image, highly magnified image of a 4-cell stage going into a morula, going into an early blastocyst and then late blastocyst. This is a real image of a human embryo growing at a very, very early stage. And this is the inner cell mass, the ICM, which is being shown here. So basically what the clinicians do is they harvest these embryos after in vitro fertilization trials are done, and the parents say that we don't need any more of these embryos, and because they've utilized all the embryos, so they don't want these. So these researchers go with a needle and they poke into this blastocyst, and they harvest or they suck out these inner cell mass cells, also known as embryonic stem cells, and they start growing in the lab and they start differentiating them. So the long story or the short point I want to make here is, if you want to harvest these cells, the embryos need to be destroyed. So this is just a cartoon that I showed you, so I'm not going to spend time on this. So one of the challenges is, what do you do with the embryonic stem cells when you harvest them? Well, you can do millions of things in the lab. The first thing you're going to do is you're going to start growing those embryonic stem cells. You grow them in a specific house, in plastic plates. You provide them different kinds of food. You provide them carbon source. You provide them nitrogen, phosphorus, sulfur, peptides, hormones, salts, minerals. You provide a cocktail, which we call growth media, and we start growing them under different conditions. For instance, in condition A, we add a chemical 1. In condition B, we add a chemical 2, and so forth. So depending on the different chemicals that you're using, these embryonic stem cells will start turning into different kinds of cells. So for instance, with chemical A, they will turn into skin. With chemical B, they'll turn into neuron, and so forth. So really, the power lies in the hands of the researchers to determine what he or she wants the embryonic stem cell to turn into. Do they want to turn them into a liver? Do they want to convert them into a blood, and so forth? So depending on that, you can grow the stem cells under different conditions. And there's about 50 different conditions that you can grow them under. And they have been worked out very intricately for the past 35 or 40 years. It took a long amount of experimentation, a lot of hit and trial to find out what works in converting these embryonic stem cells into different cell types here. So by now, you should know that embryonic stem cells are very powerful, they are very valuable, and they can turn into any cell type in the body. Now, the second kind is the adult stem cell. As the name suggests, they are not coming from the embryos, but rather they're coming from the adult body. So these are also undifferentiated cells, which are found in different tissues or different organs in the adult human body. And what is their main function? Why do we have stem cells? Even when we are fully formed as an adult, why do we have stem cells? Well, it's extremely crucial to have those adult stem cells because they are utilized for tissue maintenance and repair. Like I mentioned earlier in the talk, whenever we suffer an internal injury, whether there's a bleeding or we hit our knee against the edge of a table or whatever injury we have in the body, for us, we may not realize we have an injury because we don't have the pain, we don't see any swelling, but internally the injury is there. The tissue may have got damaged, the blood vessels may have broken. We may not realize it, but the body inside gets into full gear to start tissue maintenance and repair. That means they have to replace those damaged cells. And you don't want those damaged cells to accumulate in your body because these damaged cells need to be removed from the body. Otherwise, they will be just lying there as debris, as trash, and we don't want trash to accumulate in our body. So we have an excellent system for removing the trash from a body. That's another system which we will not talk about. But once the trash is removed, you have to replace them with good cells. And that's where the adult stem cells come into play. Once they realize that there is damage inside the body, they get signals from different proteins, and those proteins tell them, aha, we have suffered some damage here in this tissue. Please make more copies of yourself and differentiate so that we can replace the damaged cells. So these adult stem cells have been found in a variety of tissues I've listed here: the blood, the brain, the muscle, the skin. By the way, the skin is subject to so much trauma. Every single day we walk in the sun, we kind of erode our skin through various kinds of cosmetics, through different kinds of skin treatments. So we shed a lot of skin cells. So our adult stem cells lying in the skin are very, very important for replenishing our skin layers, the epidermal layer. And likewise, the bone marrow is very important, because if there were no adult stem cells in the bone marrow and somebody had to undergo radiation treatment for cancer or any other disease, then you are going to lose a lot of bone cells. So if there's no replacement, then that's going to be fatal to the body. So adult stem cells, after radiation treatment has been completed, the adult stem cells kick into big gear, and they start replacing or replenishing the lost bone marrow cells. So although I did mention embryonic stem cells are the most powerful, but one should not underestimate the power of the adult stem cells. They are absolutely crucial in our body. So like I mentioned here, adult stem cells can be multipotent, but the only problem with adult stem cells is they are kind of hard to isolate. So imagine, I just told you adult stem cells are present in the brain, but how can you harvest cells from the brain? The brain is completely protected with a very hard scalp, very hard bones. And even if you were able to slice the bone out, these adult stem cells are lodged very deep inside the brain, and it's virtually impossible to harvest them. It's easier to harvest stem cells, let's say from the liver or the kidney or the bone marrow through a spinal tap, but virtually very difficult to harvest from very deep rooted tissues. And also, unlike the embryonic stem cells, the adult stem cells have a very limited lifespan. They cannot go on, they cannot grow unlimitedly like the embryonic stem cells. The embryonic stem cells are practically immortalized in culture. That means they can grow and grow and grow and never stop dividing. But adult stem cells have a limited capacity for this viability. They may grow for maybe 50 generations, 70 generations, and then they will stop dividing. So, because the role is such that they are only needed at the time of tissue repair and damage. So, like I showed you earlier, now researchers are finding that pretty much all our tissues are actually having dormant adult stem cells lying in them, and they are activated whenever there is a need for tissue repair and maintenance. So you can see adult stem cells are pretty much in the major organs of our body. Even in our eyes, the cornea and the retina, they also have adult stem cells. So adult stem cells are multipotent, as I told you earlier, and they can differentiate, but they cannot differentiate into any cell type. They will only differentiate based on the family that they belong to. So the blood cells will make the 3 different kinds of blood cells. The stomach cells will make those 4 different kinds of stomach cells. The neuronal cells will make the 5 different kinds of brain cells, the neurons, the oligodendrocytes, the Schwann cells, the astrocyte, the glial cells. So that's why we call them multipotent. They belong to a very specific family type, and they only resort to that family type. So what are the big questions about adult stem cells? So we know a lot about embryonic stem cells so far. We know they are pluripotent, they are very fast growing, and we also identified the proteins that they produce. But there are a lot of questions that we still do not know about adult stem cells. For instance, are they present in every tissue in the body? Are they lying dormant? Are they sleeping? And do they need to be woken up? How are they formed in the body? Are these leftover stem cells coming from the embryos, or are they new cells which are produced in the body? And can they be turned into the different cell types based on simple signals, or do they have to be provided very complicated signals? And the most important, in my opinion, the most important question is, what protein molecules or what signals are needed to give them a command and tell them that it is time to make new cells because a certain portion of the body has been diseased or injured. So what is this signal which is coming, and who is giving that signal? So apparently that is a major question, because if we can understand that, and a lot of researchers have actually worked on that, and they have nailed down a few signals, but there's still a lot to be explored in adult stem cells. The third is the cord blood stem cells. And as I'm sure all of you know, cord blood is basically the blood which is found in the placenta or umbilical cord, which is joining the mother's nutritional system with the growing fetal nutritional system. And through the placenta, the mother is able to provide all the nutrients to the growing embryo inside her womb. So a very important connection. And cord blood stem cells are found in the placenta, and they are hematopoietic in nature. Hematopoietic means giving rise to blood cells. And these cells are found in a very small number. That's the only disadvantage. They're found in a very small number, only about 1 to 1.5% in the placental cord, but they have been harvested and they have been triggered in the lab under certain conditions to turn into different cell types, particularly the neuronal cell type. So they have been utilized. Excuse me, one moment. My apologies. I forgot to mute my phone. So these cord blood stem cells have been turned into neurons, but under very difficult conditions. First of all, they are found in very small numbers. So once these cells are harvested from the placenta, they have to be expanded. By expansion, I mean you got to make more of them before you can actually start turning them into neuronal cells. And of course, these are valuable, but since they are so small in the cord blood, not very many people use it because it's a very large and a cumbersome task to make more of them because they don't grow as well in the lab. But they can be used to treat a number of immune related genetic diseases. And that is why a lot of women today, at the time of childbirth, opt for preserving their placenta. And that placenta, after it's severed from the baby, is actually frozen down and stored in at a very ultra cold temperature, minus 80 degrees. And depending on the need to revive these stem cells in case there is a genetic disorder in future subsequent children of that couple, they can actually utilize these stem cells to generate normal cells and do the stem cell transplantation on their subsequent children. So this is why several of the ladies actually start preserving uh, their placental cord, and even today it goes on quite a bit here. So this is just a graph which shows you what the placenta looks like. So this is a very robust, it's almost like a rope which is carrying a lot of blood and nutrients from the mom to the baby. And once it's severed, then this is frozen, or the cells can be isolated and frozen depending on which medical facility one is getting the child delivery done. And of course there is a cost for it because they charge you for preserving, and I'm not sure what the cost is, but I think Kaiser was charging several years ago $500 a year, and I'm not sure what the cost is right now because they have to be frozen at ultra low temperatures, and those freezers suck up a lot of energy, and hence the cost actually adds up. So the fourth category is amniotic fluid derived stem cells. And this is also tied to the growing embryo in the body because the amniotic fluid is the fluid which cushions the growing embryo inside a womb. And this fluid is very important because it provides a very soft and a cushioning effect to the growing embryo, and also contains lots of nutrients which the embryo imbibes. And again, there are certain stem cells found in the amniotic fluid, and they have been differentiated into the liver and neuronal lineages. But because they are present in such a small number, only 1 to 2%. Again, very few researchers actually work with AF-derived stem cells. So of all these four categories, the one which is used extensively is either adult stem cells or embryonic stem cells. But now, with advent of newer technology, which garnered a Nobel Prize several years ago, we don't even need to use embryonic stem cells. We can use what we call induced pluripotent stem cells, which I'll talk about later. So this is just showing you an image, which I just forgot to show you. This is how the amniotic fluid is harvested and the stem cells are removed from those amniotic fluids. And by the way, it's a very risky process because someone who is extremely skilled has to be able to do this, because the risk or the danger of damaging the embryo stands strong here if someone is not doing it properly, because you don't want the needle to pierce the embryo. You just want the needle to stop at the amniotic fluid and harvest the fluid. And this is amniocentesis is routinely done right now because the amniotic fluid contains cells coming from the embryo. So amniotic fluid is harvested in the first trimester, or sometimes even in the second trimester, to see whether the cells of the baby are exhibiting any chromosomal aberrations or genetic defects, which can guide the future line of delivery for the couple. So harvesting amniotic fluid has been done through amniocentesis, but harvesting stem cells from them has to be done very, very carefully. So the whole million-dollar question is, if we are working with embryonic stem cells, and there is absolutely no doubt that embryonic stem cells are extremely valuable, they are very powerful, they can be converted into any cell type. So where do we get these embryos from? Well, most of the embryos are coming from fertility clinics. So fertility clinics are frequented by couples who are either not able to conceive, or they're able to conceive, but they're not able to carry the pregnancy full term. So essentially, the process which is used is known as IVF, in vitro fertilization. As the name suggests, in vitro means outside the body, in vivo means inside the body. So in vitro fertilization means the fusion between the sperm and the human egg is happening outside the body. So basically, the zygote that is formed is being formed in the lab in a petri plate. And for someone who goes for an IVF trial, they don't actually form embryos which are only like 1 or 2, because the success rate is only about 20 or 30%. So basically, the embryologist recommends that they be at least 12 or 13 embryos, because out of those 12 or 13, only maybe 3 or 5 will survive. So of those 12 and 13, the embryologist screens for the most healthy embryos, and amongst those healthiest one, they may implant either 1 in the woman's uterus, or 2 to generate twins, or even 3 to generate triplets. But the risk increases as you implant more and more embryos. So the long story is, if you have 13 embryos and you utilize only 2, what do you do with the remaining 11? So that's a big question. So the couple is planning to have more babies in the future. They can let the fertility clinic know, okay, freeze down my embryos. Maybe 3 years down the line, I may have another child. Okay, but nobody's going to have like 13 children or 13 implantations, because that's very risky. So even if a couple has 4 children, then what do you do with the remaining 7 embryos? So those are known as leftover embryos. And what do you do with these embryos? Some of the embryos are normal. That means if implanted, the children would be absolutely normal with no genetic defects. Some embryos are not implanted because they could have chromosomal defects, they could have genetic aberrations. So what do you do with all of this? So regardless whether the embryo is normal with a clean genome, or whether the embryo has genetic defects, the whole problem is, what do you do with these leftover embryos? So that's where the whole ethical concern comes. Should we utilize these leftover embryos, or do they belong to the couple, or should we let them sit in the freezer, or should we give them to the researchers to isolate the stem cells and study how they are growing? So what do we do? So there's a huge ethical sword which lingers on our heads for legislators, for the public, for the parents, for the doctors. What are we doing with the embryos? So if there is such a big debate about embryonic stem cells, then why do we not use adult stem cells? Why even go for embryos? I already told you there are lots of adult stem cells in the body. That has already been proven, but why don't we use them? Well, there are two reasons why we don't use them. One, they are not as plastic as embryonic stem cells. So here the term plastic does not mean, the regular thick plastic that we use in our homes. Plastic here means the capacity to turn into any cell type. That's why we call it plasticity. So adult stem cells are not as cool or as plastic as embryonic stem cells. They will turn only into a specific kind of lineage. They will not turn into any kind of stem cell. So that's one drawback. And also, the existing adult stem cells have very limited genetic diversity. And we don't want any limitations in our genetic diversity. We want a more comprehensive and a broad genetic diversity to be able to understand the differences between different individuals. So because of these two reasons, primarily the number one reason is why we do not resort to using adult stem cells extensively in the lab. So coming back to the embryos, what do we do? So the proponents of embryonic stem cell research say, hey, we got hundreds and thousands of these frozen embryos sitting in the fertility clinics in all 50 states, and eventually they'll be destroyed. They're not going to be preserved forever. So eventually, once the command is given by the parents, because the parents own them, it's their property. So they have to sign off on a consent saying that, okay, we agree for these embryos to be destroyed. So the proponents say, these frozen embryos are sitting there and they're not doing anything, and the parents don't want them because they already have 2 or 3 children. So what are we doing with these embryos? So why don't we use them for research? Why don't we use them to find out how embryonic stem cells work, how they differentiate, and we can probably work on finding some cures for diseases, because eventually they're going to be destroyed. The opposite side, the opponents say, hey, no, we cannot destroy them because embryos are life. And these embryos, which are not being used by the biological couple, they should be given for adoption to couples who are not capable of conceiving. So both points are correct. Actually, both points are great. One, if there are thousands of embryos, do we have enough couples who want them for adoption? And likewise, for infertile couples, these embryos are a blessing because they can adopt them. So I'm not saying that either proponents are correct or opponents are correct. It's an open debate because both points are correct at this point. It's a question of how many frozen embryos are there and what kind of biological information we can get from them, which can benefit the society in terms of finding cures for future diseases or even current diseases. So potential uses of stem cells are too many to list here, but I've listed predominantly 3 of them. One is by growing these stem cells in the lab, we can monitor how they develop. We can expose them to new drugs, and we can see how they respond to different drugs. And most importantly, today, this is a very hot area, is we can grow these cells and test them for cell-based therapies. For instance, we can grow cells coming from an Alzheimer's patients, and we can treat them with different genetic manipulations or different drug manipulations and look at the formation of the beta amyloid proteins or the tau proteins in vitro in the lab and see how cell-based therapies can be developed, particularly to fix very, very serious diseases like Parkinson's or Alzheimer's or ALS or multiple sclerosis, and you name it, diabetes, arthritis. All these diseases can be studied in the lab, and they are being studied in the lab extensively to develop cell-based therapies. And that's why the use of human stem cells is very important in the lab currently. Otherwise, we will not be able to analyze what works and what does not work in the lab. That means if we don't do lab work, how can we bring something into the market? All the basic research has to be done in the lab before we can start applying it. So for transplantation purposes, and this is routinely done, particularly in cancer patients where the bone marrow is actually destroyed because of the radiation treatment. And so the stem cells or The blood cells need to be replaced very, very fast to generate the immunity in a cancer patient. So for transplantation purposes, stem cells should be able to divide very fast and extensively. They should be able to generate large quantities of tissues. They should be able to specialize into the desired cell type. And most importantly, once transplanted, they should be able to survive in the recipient. They should not die off like after a week, otherwise, it's a waste. And they should be able to talk with the surrounding tissue. The surrounding tissue is made up of collagen, extracellular matrix, muscles, different kinds of blood vessels. So they should be living happily with their neighbors and not cause any kind of immune rejection. They should function properly, and basically, they should not cause any harm or any risk to the recipient. So, transplantation is not an easy process because once transplantation is done, there is always a risk of rejection by the recipient body. And that is why transplantation can be a very, very tricky process. So, where can stem cell research help? Well, lots of diseases. I've listed a partial list of diseases: heart disease, immune disorders, diabetes, bone disorders, cancer, neurological diseases, birth defects, you name it. There's lots and lots of people suffering from these diseases in the US and in the entire world. And people can benefit uh, from the results coming from stem cell research at this point. So the first ethical hotspot, which I've already discussed here, is should we or should we not use human embryonic stem cells? That is a very, very big concern at this point. And the second ethical hotspot in research, which I probably won't have time right now, it's 10:52, is should we allow cloning? Should cloning be allowed in humans? That means basically, I'm going to explain to you in very, very simple words, is you take a male sperm, you take a female egg, you fertilize it outside the body in a plastic container in the lab, you form a zygote. And once the zygote grows a little bit bigger in about 12 or 14 hours, you start genetically manipulating that zygote. That means you go and start changing the genes in a very, very small manner, not major changes. Maybe scan the genome to see if there are any genetic defects that may happen down the line in the embryo. If yes, can you fix those genetic defects? Which is great, by the way. If you can fix a genetic defect, then the child which will be born is normal. And then you can implant that fixed embryo back into the mother. But this can be misused. And that misuse comes from the term known as reproductive cloning, where couples may opt to genetically alter their embryos and their children to change their skin color, to change their eye color, to change their hair color, to make them have more muscle mass. So these are cosmetic changes. And so they're not required to fix a disease. But this reproductive cloning is not allowed currently because that can have major, major, major ethical concerns. Because the moment you change the sex cells in a growing embryo, that is a permanent change. And let's say if the sex cells, or the DNA of the sex cells is changed in the growing embryo, and the embryo is implanted back in the mother, and she goes into full-term pregnancy and she delivers a child. Now that change is permanent in that child. And when the child grows older and has children of his or her own, that genetic change is transmitted for the next million generations. And that is why reproductive cloning is prohibited currently. It is banned right now, but somatic cell cloning is allowed. Somatic cell cloning means fixing the non-reproductive cells in the human body. And so the genetic change happens only in that human being. It does not get transmitted down to the children because it is somatic. And when the person dies, those cells die with them. But in reproductive cloning, those engineered cells are passed down to the progeny. And that is why that is a second ethical hotspot here. And then we've already discussed the embryonic stem cells. So there's a whole lot of issues, You have to get informed consent from patients. You have to protect the human subjects. You want to make sure the genetic intellectual property regulations are followed. People can actually start doing all sorts of very weird research. They can start combining a human cell with an animal cell, and they can make hybrid cell, which is almost like, unheard of, and that should never ever be done, because humans and animals are very distinct. So even if you make a hybrid cell, and what kind of features does that hybrid cell have? So there's all sorts of very, very unscrupulous researchers there who can start conducting different kinds of research, which should actually not be allowed. So there's a whole lot of issues with embryonic stem cells here. So we have mechanisms in place. We have agencies within the US which are very strict, which are very regulatory. There is legal reinforcement. There's protection for the subjects who are donating the embryos. There is protection for the subjects, for the researchers who are conducting research. So, but education is the most important principle here. We have to educate the public. We have to have an open dialogue with the public, as we're doing right now, to explain to the public what are stem cells, why are there so many issues, what is beautiful about them, what can they be used for, and so forth. So, without proper education, there's a lot of blind information which is floating around. So I'm going to stop here, because somatic cell nuclear programming is a very cool area in itself. But right now, I actually want to talk, I want to give a baton to my students who will be talking about stem cells and their use in diseases here. So I'm going to stop sharing at this point. And I will pass on to the first group, who is going to talk about the use of stem cells in cardiovascular diseases. So who is group one? Can you please identify yourself? Shrija, yes, please go ahead. So these are our 8 or 9 stem cell students who will be starting their stem cell internship in about 2 weeks' time. They have been placed in different areas within California, in different labs: Stanford, USC, Cedars, Children's Hospital of LA, SOC, Scripps, UCs, and so forth. And they have formed themselves into 3 groups, and they will be doing a 20-minute presentation each, discussing the role of stem cells in fixing different kinds of diseases, cardiovascular disease, blood cancers, and so forth. So the first group is Jamima, Shrija, and Kumari, and it's all yours. Please go ahead. And if you can expand your screen a bit, yeah, that's great. Thank you. Go ahead.
Hello, everyone. Good morning. My name is Jemima Moses, and I'm a master's student at Cal State Channel Islands, and I'm also the CERN Bridges intern for this year, and I'll be joining Cedar Sinai pretty soon for my internship. And today we are going to talk about, me and my team are going to present about how stem cells can be used to treat cardiovascular diseases. So let's start with a little bit about what are cardiovascular diseases. Can we go to the next slide, please? So cardiovascular diseases are a group of disorders, and they are most commonly referred to as heart and blood vessel diseases. And these diseases involve malfunction or damage of the heart, blood vessels, and sometimes the entire circulatory system. Cardiovascular diseases can range from mild to severe and can have serious implications for overall health and well-being. And some of the most common types of cardiovascular diseases include acute myocardial infarction, which is often known as heart attacks. And the second is congestive heart failure. So let's move on to the next slide to learn a little bit more about how does a normal heart work. I'm sure everyone is aware about this, but I just wanted to reiterate. So heart is a fist-sized muscle that pumps blood to almost all the parts of our body. And fun fact about heart, it beats ever since that we are born till the time that we pass away. And each day our heart beats around 100,000 times, and it continuously pumps about 5 liters of blood throughout our body, which is around 8 pints. And when we look at the anatomy of our heart, It pretty much is divided into two chambers, the upper and the lower chambers. And the upper chambers are called the left atrium and the right atrium. And the lower chambers are called the left ventricle and the right ventricle. So primarily the right side of our heart, the one which is depicted in this image as blue color, it receives the deoxygenated blood from different parts of our body, and our heart pumps it to the lungs where it is purified or where oxygen is added to our blood. And the left side, which is depicted in a red color here, it receives the oxygenated blood from the lungs, and it is pumped back to the different parts of our body from the left side. So there are three major muscles in our heart. The first is the pericardium, which is the thin outer layer which surrounds and protects our heart. And then there is myocardium, as you can see this pink separation between the two ventricles. It is a thick muscular middle layer that contracts and releases, which helps the heart to pump the blood. It basically pumps the blood through these contractions and relaxations. And then the third type of muscle is the endocardium, which is a thin layer that makes up all these linings inside the chambers and the valves that we see here. And moving on, we will discuss about different types of heart diseases. So if we could go to the next slide. Yeah. So the most common heart disease, which is the major contributor for any kind of cardiovascular disease, is the coronary artery disease. And it occurs when the coronary arteries that supply blood to the heart becomes narrows or thickens due to deposits, fatty deposits like these that you see in yellow color here. These are also called plaques. And we also see about heart failure. Heart failure is the next most common type of heart disease, which is when the heart is unable to pump blood effectively to meet the entire body's needs. And it can result from various reasons. One of the major reasons being high blood pressure or damage to the heart muscles. And then there is cardiomyopathy, which is thickening of the myocardium. And it just makes it harder for the heart to pump blood. And it can be caused by various factors, sometimes infections, or it can be genetic, and it can also be caused by certain medications. So now, if we move on to the next slide, we will discuss a little bit about the symptoms of heart diseases. These are very common symptoms that most of us often ignore, and these are not considered as something that can be life-threatening. For example, chest pain, discomfort, shortness of breath, dizziness, or exhaustion can be sometimes taken as, oh, we just need to get a little bit of rest and we'll be fine. These are the most common types of symptoms that are often ignored. Now, let's move on to the next slide, where we will discuss about how cardiovascular diseases are the world's #1 killer. They are the cause for death of around 18.6 million people every year, and it contributes to 33% deaths all around the globe. Now, next, move on to the next slide, where we can clearly see that cardiovascular diseases remain the leading cause of death everywhere in the world. And according to a report from American Heart Association, just in the United States, cardiovascular diseases were the main reason for about 900,000 deaths in the year of 2020. And another interesting statistic that I found from this report was that one person dies every 33 seconds in the United States from cardiovascular diseases. I'm going to let that sink in. That's a large number. And coronary artery disease is the most common type of heart disease, which is killing almost 300,000 people just in the year of 2021. Now, I would like to invite my friend Zeel to continue the presentation. Thank you.
Pathophysiology of the heart disease. So the pathophysiology of the heart disease which involves the complex interactions between various physiological processes, which due to which there are like some cellular changes and the changes in the molecular mechanisms. And here are some of the key factors and the processes which include the atherosclerosis, in which the coronary arteries, that is the coronary artery disease. The atherosclerosis is a central process, and in that there will be the accumulation of the fat deposit, also some other substances within the wall, and over a time it will the deposits will harden, and the arteries will get narrow, and which will limit the flow of the blood. After that, the second the second is the endothelium dysfunctions, in which the blood vessel inner lining will normally helps to regulate the blood flow and the vessel tone, and due to the high pressure, due to the high blood pressure, high cholesterol levels, smoking, it can lead to the impaired blood vessel function. Inflammation also plays a crucial role for the development of the heart disease, and due to which there will be a damage on the inner lining of the blood vessel, that is the endothelium, and it will promote the formation of atherosclerotic plaques, and due to which the inflammation will happen and the cell will release some kind of substances that will contribute to the plaque and instability. Then the hypertension also causes the heart failure. That is, the heart will unable to pump effectively to meet the demands. The myocardial infarction, that is the heart attack, which is due to the coronary artery when it is blocked, and when the blood clot inside it, and the blood supply and the portion of the heart muscle, it gets cut, and which will result into the cell death and affected area, and due to which there will be a heart attack. Or there are also some of the genetic factors and the diabetes which involves in the for the heart to dysfunctions. Next slide, please. There are also some of the causes for the after the heart attack. There are some of the things which is due to which causes the heart attack, that are the recovery period, medications. There are some of the medical treatments, then due to the changes in the lifestyles, the recovery, which includes the ECG, that is the electrocardiogram blood test. Then there should be an immediate treatment, recovery in the hospital for the complications, and it may receive that is to prevent the blood clot. Then another one is the cardiac rehabilitation. Medications, there are some of the medications which are given to the experiencing a heart attack, and the patient may feel anxious, depressed, fearful, and adjusting to the change, and it will adjust to the changes in the lifestyle and the death. So like aftermath of heart attack, the heart attack was one point I have to add in this is that during the COVID-19 period also, the doses which were given to the patients was also considered as one of the reason for the heart attack, such as due to the cytochrome storm, which increases the level of the cytokinins in the patients. Next slide, please. So here are the current treatments through which the heart diseases can be cured. So here the medications are given such as the statins, and these drugs helps to lower the cholesterol levels and reduce the risk of the atherosclerosis and the heart attack, that is clotting of the blood and narrowing of the coronary arteries. Then second will be the the second drug, that is the beta blockers. Then there are also some ACE inhibitors which will and which are used for the high blood pressure and certain blood conditions, and antiplatelet, anticoagulant medicines. But then heart failure management, but there are no other treatments. Ahead of this. There are some advanced treatment like invasive procedures in which the conditions are treated with minimally invasive techniques such as like aortic valve replacement, aortic stenosis, regenerative medicines for the repair damaged heart tissues. But there were some side effects and are not completely cured. And they were having some kind of disputes. So yeah, it's the current treatments are still challenging. So there is the ultimately the thing is they have to go through the surgery or take the medicines which are having certain side effects. So I'll pass over to Shrija to continue this.
Thank you, Zil. Hello all, this is Shri. Can you all hear me? I'm getting echo, so.
Yes, now we can hear you fine.
Yeah, thank you. So what are stem cells? As Dr. Parmar mentioned, so stem cells are cells that has the ability to develop into various cell types. So they have this unique characteristics like, in tissue repairing and regeneration. So this makes them a potential cure in treating various diseases, in our case, cardiovascular diseases. So let's see the types of stem cells. They can be classified based on their sources and based on the differentiation potential. So based on the sources, they can be derived from 3 to 5 days old embryos that are embryonic stem cells. And it can also be derived from inner cell mass of blastocysts, in other words, a 6 days old embryo. And they are called fetal stem cells. And then we all know there is an umbilical cord stem cells, and also it can also be derived from the adult stem cells, adults. So for example, there's hematopoietic stem cells. They can differentiate into all blood stem cell types. And there are mesenchymal stem cells. They can develop into cardiac cells. Based on their differentiation potential, they can be totipotent, pluripotent, and multipotent. Totipotent is the ability to develop into a complete organism. Now, we all came from zygote, right? The fertilized egg, that single cell, one cell embryo that can develop into an organism, that is totipotent. And then pluripotent is the stem cells that can give rise to all cell types. Like they can differentiate into blood cells, neural cells, neurons, heart cells, and so on. And the multipotent stem cell, they can develop into a family of cells. If it is a blood stem cell, like the name is hematopoietic stem cell, they can develop into all blood types like WBCs, RBCs, red blood cells, platelets, and so on. So stem cells in cardiovascular therapy. So stem cells, are like stem cells derived from bone marrow, endothelial progenitor cells, mesenchymal stem cells, cardiac stem cells, progenitor cells, and iPSCs, induced pluripotent stem cells. They all can protect the heart by formation of myocardium or increased resistance against apoptotic stimuli, which means apoptosis means cell death, programmed cell death, and neoangiogenesis, which means by developing new blood vessels, and so on. So here in terms of ischemic heart disease, where the heart is not getting enough oxygenated bloods, then at that time stem cells can be used to stimulate the new blood vessel formation, and it will improve the blood flow to the heart. And in case of heart failure, it is able to regenerate the damaged heart tissue and enhance the cardiac function. And in cardiomyopathy, it can be it has the potential to replace the damaged heart muscle cells. So how are we gonna deliver these stem cells into the heart? By intravenous infusion, which means stem cells are sent directly into the veins using a needle or tube. And the issue here is, there could be a challenge of like poor homing, and then intracoronary injection, which is directly injecting the stem cells to the coronary artery. And there could be an issue with a block, or it could the stem cells can be washed out, which we have to study more on that. And then surgical implantation, implanting the stem cells by heart surgery, which involves incision. Yeah. So thank you all for your attention. If there is any questions, we are happy to answer. Thank you.
If any of you have any questions, you can ask the students now or wait till the end, whatever works for you.
What is the most common type of therapy happening? Which of those three types of implantations of the stem cells is the most common?
I think it is the intracoronary infusion, because it can directly inject into the affected place, right? The coronary artery.
And are researchers finding that the embryonic stem cells do the best work in this, or would the adult stem cells, because it's specific to blood, work just as well?
Let's get it right. I think mesenchymal stem cells can work best, because or cardiac stem cell progenitors, they can differentiate directly into the next step is the cardiac cells, right?
So Darcy, to answer your question, it's going to be mostly the CSCs. Go back to the slide, Shrija, the previous slide, the one that you just showed. Yeah, this one. So we have the cardiac stem cells. Those would work the best, because for embryonic stem cells, if you were to inject them, I mean, the embryos are coming from a separate person, so there could be immune rejection if you convert them. But essentially, if we are able to harvest cardiac stem cells, which again can be quite problematic if the heart is damaged, those cardiac stem cells, the CSCs, would be fine. But the best option would be the iPSCs, which unfortunately I did not have the time to talk about, the one on the right here, induced pluripotent stem cell, where adult cells, not stem cells, adult cells are taken from your body and they are reprogrammed and converted back into an embryonic stem cell-like stage, and then redifferentiated into cardiac stem cells. And then those cardiac stem cells are transplanted back into the heart. So it's sort of like a long process, but because your original cells are coming from your own body, there is absolutely no question of rejection. So there's various mechanisms. I think it's just a question of efficiency, efficacy, and what works and what does not work. So it's still, I would say, a playground at this point.
So
if you wanted to read about that last process, which I guess is the basis for the Nobel Prize, could you give us a good reference?
Certainly, I will definitely send the reference to either Michael or Daniel, and they can share with the Oli group.
Super, thank you.
Sure. If there's no more questions, we can move on to our second group, who would be discussing, I believe, leukemia. Please load your presentation, group number 2. Thank you so much, group number 1.
Just a moment, Dr. Pam. We'll be just sharing our slide.
Certainly.
One second, Dr. Parmar. We're just facing a little bit of technical issues.
Dr. Palmer? Can I add to that request that last
Certainly.
slide that gave the references?
Correct. Would you like those references?
Yes, ma'am. If I could just send the slide out, we could go from there.
Sure, sure. I'll request the students to send me that particular slide, and then I'll share that.
Super, thank you.
Sure.
I hope it's visible to everybody.
It says loading. So what I suggest is don't play it in the presentation mode, because that's where I also faced a problem. Just play it in
I
the regular
see.
PowerPoint. Yeah.
Okay.
Expand the screen. Yeah. The same thing happened to me. So I don't know
Oh.
why. Just make this bigger so we can, and let go of the left taskbar.
Mhm.
Minimize that. Yeah. And expand this a bit more.
Okay. So I hope everybody can see the presentation, and I'll start with presenting it. So hi, everybody. My name is Sahitya, and my team members and I will be talking about stem cell therapy in leukemia. Before talking about how the therapy itself works, I want to talk more about what leukemia is and why is it important to have various options while treating this disease, with stem cell therapy being one. Next slide, please. Okay, so these are the different contents that we're going to talk about. So I'll be talking about leukemia. Cypriya will be talking about what the stem cell therapy actually is. And Raina will be talking about how we can incorporate this stem cell therapy in leukemia. Next slide, please. So what is leukemia? So leukemia is a type of blood cancer that originates in the bone marrow. Since it originates in the bone marrow, it affects both the marrow and the blood cells. If you remember Dr. Parmar's presentation, she was talking about multipotent cells. So one of the multipotent stem cells, called the hematopoietic stem cells, originate from the bone marrow, and they help in, so they create the different types of blood cells that we have: the red blood cells, all the types of white blood cells, which when I mean all the types, I mean the eosinophils, basophils, neutrophils, monocytes, lymphocytes, all of these. So when it affects the bone marrow, where it's technically affecting mostly the white blood cells, and that's why it is really dangerous. And it warrants our attention, and we need to treat it. So at its core, leukemia involves an uncontrolled proliferation of the white blood cells. Like I've put up a picture which shows how normal blood would look under the microscope, and how leukemia, a person with leukemia would have its blood will have their blood under the microscope. So you can see that there's a significant increase in the white blood cells over there, whereas the normal blood does not have many. It just has like the allowed amount of white blood cells, if you know what I mean. So it is so white blood cells are very important for our immune system. They are the ones that keep the infections or any foreign particles out of bay, and they protect our body from harmful substances. So these cells play a very important role in safeguarding our well-being. And if the lymphocytes or the white blood cells growth itself spirals out of control, it gives rise to this very complex condition called leukemia. And so one of the things that you can characterize leukemia with is the increased white blood cells, like I'm repeating at this point. So this cancer can be categorized into two types. The first type being acute, meaning that it progresses very fastly, and it is most commonly seen in children and adults. And the second type is called chronic, which means that it progresses relatively slower, and it's most commonly seen in adults. And so again, this cancer is broadly categorized into four types. So they are called acute lymphoblastic leukemia, which means that it's a fast-growing leukemia that affects the young and mature lymphocytes. It is most commonly seen in children, which is really sad, but mostly all the children, majority of the children are seen to have leukemia with this specific type of leukemia, and it is it progresses really rapidly, and that's why an intense treatment is really important. It should be fast, like you need to like find it out really fast and start with the treatment fast because it's very aggressive. The second type is acute myeloid leukemia. Again, this is also a fast-growing leukemia, but it affects the myeloid tissue. It's kind of like a progenitor for these lymphocytes. So it's not a developed mature lymphocyte, but it's kind of like a premature, the immature cells. So this leukemia affects these type of cells. And again, this type of leukemia is seen both in children and adults. And the treatment involves chemotherapy and targeted therapy. I will be talking about what sort of therapies are there later on, but right now I just want to explain the different types of leukemia conditions we most commonly see. So the third type of leukemia is chronic lymphocytic leukemia. So like the name suggests, it's chronic, meaning it is slowly developing, and it is mainly found in older adults, and the treatment varies based on the disease progression, the patient's health, sex, age, all of that. And the last type is the chronic myeloid leukemia. Again, it's a slow growing, but it affects the immature cells, the myeloid cell growth. And it's typically, again, seen in adults. And targeted therapy is the primary treatment, unlike the others where chemotherapy is the primary treatment. The next slide, please. So who is affected by leukemia? There are diverse individuals that are affected. We cannot specifically say like, oh, this is like how breast cancer is. Like we can tell that breast cancer affects women more than men. But here leukemia affects diverse individuals spanning all ages. However, it is most frequently seen in adults and older individuals. Again, I'll present the demographics in the later slides. But just because it's more common in adults and older individuals doesn't mean that it doesn't affect children. Like I mentioned previously before, the acute lymphoblastic leukemia is the most common type of leukemia you see in children. And I've also mentioned what sort of symptoms you need to look out for when it comes to diagnosing leukemia. So there's going to be weight loss, frequent fevers, infections, and the infections is because leukemia affects the immune system, the white blood cells, and that's why the immune system weakens and we have frequent infections. There's going to be easy shortness of breath, weakness, pain, and tenderness. Your spleen is enlarged, which is a lymphatic organ. It is involved in the immune system, so there's effects even in that. So all of these are common symptoms when it comes to leukemia. And so there is like a person's lifestyle is one of might be one of the reasons why a person can develop leukemia over the time. But apart from that, there are also other risk factors when it comes to this. This includes if a person is exposed to high levels of radiation, any chemicals, and if there's a family history of leukemia, and there's any genetic disorders such as Down syndrome. All these factors also play a really important role with regards to the development of this disease. Next slide, please. So what are the treatment approaches for leukemia? So when we talk about treatment in leukemia, it's not one size fits all. So it has to be carefully curated and tailored based on the individual. Like you need to check what my age is, what my sex is, what race I am, ethnicity, and if I have like any other condition. So everything should be considered before giving a treatment for this disease. So the type of leukemia, its stage is also very important when you're crafting an effective plan for the treatment. Among the plethora of options, chemotherapy stands at the top. Like you've seen, like most of the types of leukemia, the majority of the patients are treated by using chemotherapeutic agents. But apart from chemotherapy, you also have other types such as targeted therapy, where it targets the specific cell. Well, in chemotherapy, your whole body is affected. Like it's not targeted. It doesn't really target only the cancer cells. It targets all the cells in your body. And that's why there are really, really bad side effects when it comes to chemotherapy. But there are other therapies too, such as targeted therapy. And another type is radiation therapy, stem cell therapy, which we'll be talking more about later on. And there's another type called immunotherapy. So there are different options, and it really based on how well you can handle it and if it works the best for you or not. Next slide, please. So I've given like the base, the different types of treatments and what sort of drugs do they use. The first one is chemotherapy. So like I said, it targets all the cells. It's not really specific, but still it is the most prioritized. Like it's like super famous. When it comes to cancer treatment, everybody first look towards the chemotherapy, and then if that doesn't work, they go towards the other forms. So cytarabine, daunorubicin, all these are different types of therapeutic agents that you can use when treating leukemia. And targeted therapy is more like these drugs are they target the cancer cells, or they target certain molecules that are involved with the cancer development, and they kind of minimize the harm that is given to healthy cells, like in case of chemotherapy. And the other types of therapy is immunotherapy. So this is where you use monoclonal antibodies. A little context is that monoclonal antibodies are created in a lab. So they work like antibodies that are already present in our body. So they act like that, but they kind of go and attach themselves to these cancer cells. So what happens is, since cancer cells are our own cells, the body, our immune system cannot really recognize that, oh, this is a cancer cell. They just see that it's your own cell, and it thinks that, oh, okay, it's normal, it's our cell. It's not something foreign. There's no need to take action against it. But they don't know that it's a cancer cell. So what these monoclonal antibodies do is that they go and attach themselves to the cancer cells, because no matter, even if it's from your own body, there are certain markers on the cancer cells that make them different. So these monoclonal antibodies attach themselves to the cancer cells, and this makes easier for the healthy, normal white blood cells to see that, oh, there's something wrong with this cell. We need to attack them and kill them. So that's how this mechanism happens. And that's why immunotherapy is also like really, really potent. It's very targeted. So that's one type of therapy you can use. And the other type is tyrosine kinase inhibitors. So they inhibit the proliferation of cancer cells itself. The next slide, please. So here is the demographics I was talking about previously. We can see that in people with leukemia, there's a 5-year relative survival. And in 2023 itself, we have seen approximately 60,000 new cases. And we can also see that among all types of cancer, 3% of them are leukemia. And when you see the death rate, we can see that 3.9% of a percentage of all the cancer deaths are due to leukemia. And if you see the trend below the graph, we can see that the death rate is slowly decreasing, obviously, because the science is being developed and there's new technology, the research is happening, clinical trials are happening, and we have more and more options to treat this cancer. So it's bad, but we're looking in the positive light that more and more research, with more and more research happening, we might be able to overcome this really horrible disease. Next slide, please. So this is kind of an overview about which ethnicity and what sex is more vulnerable to this disease. We can see that in both the male and female, both these cases, we can say that non-Hispanic white people are more vulnerable than others to this disease. And we can also see that leukemia is also frequently diagnosed in people with the age groups of 65 to 74 years old, and the median is 67. So it's not like we're saying the cancer does not affect others, but these people are more prone, more vulnerable to this to the disease, and we need to make sure that none of the symptoms or any troubles that we have in our body don't go unnoticed. We need to make sure that we kind of make note of everything and consult before it gets to early because cancer in its earliest stage is most definitely treatable. So I'll pass on the stage to my friend Cypria. She's going to talk more about the stem cell therapy.
Am I audible? Okay. So now I'm just going to talk a little bit about stem cells. So we've already been listening about stem cells since the beginning from Nitika, Dr. Parmar, and from the other presentation as well. So I'm just going to talk very briefly. So we have two types of stem cells. One could be embryonic stem cells and the other could be adult stem cells. So embryonic stem cells are basically from embryos, and now right now for research we either use unused embryos from IVF, or we have people who generously donate embryos to research, so we use those embryos for stem cell research. We also have the other type of stem cells, which is the adult stem cells. Adult stem cells can either be one from fully developed organs like your bone marrow, skin, brain, etc. But the issue is that from these stem cells, you can only form certain types of stem cells. Certain types of cells. You cannot form all the types of cells. So what science has found out later stages is that you can induce these adult stem cells to become something like an embryonic stem cell. So that is what is induced pluripotent stem cells, where you take a normal cell and then you revert it back to its embryonic stage, where it's able to produce multiple different types of cells, and then you target the type of cell that you require, and then you differentiate them. So this type of stem cells are called induced pluripotent stem cells, and they are very similar to embryonic stem cells. So the ethical issues that we were talking about, where you'll have to where there are more issues when it comes to embryos, is completely negated when it comes to induced pluripotent stem cells. So that's one plus point when you're using induced pluripotent stem cells. So there are a few questions that might be there among us. One is, what diseases can actually be treated with this stem cell research? So theoretically speaking, we can cure most of the diseases with stem cells. So that is what we know about stem cells, and that's what we hypothesize. But we still have the research ongoing, and we still require multiple proofs and methods for us to make sure that this becomes practical. And the first ever stem cell that was used in a clinic was blood forming stem cells, which was for blood cancer like leukemia. And will there be any immune rejection? So this is very similar to tissue transplant and organ transplantation. So one more question that's very common in the public is, will there be immune rejection? So yes, there can be immune rejection if you're taking it from an embryo, or if you're taking the source cell from another adult who's donated the stem cells. But if you're taking your own cells and you're inducing it to become pluripotent stem cells, then they are going to have the same genetic makeup and everything. So the immune rejection in induced pluripotent stem cells is very low to none. But there could be some when it comes to embryonic stem cells or adult stem cells from a donor. So there's also another question, which is, so let's say there are cancer cells. So cancer cells also have the same genetic makeup as the cells that are surrounded by them, like the healthy cells that are around them. But still our immune system has this ability to target these cancer cells and kill them in the initial stages. So there is another question that's around the community, whether the immune system will still be able to target your induced pluripotent stem cells because they are a little different, or will they be able to just let them slide? So this is also one of the questions that's around. A few stem cell research that's ongoing right now would be with different kinds of stem cells. So number one would be embryonic stem cells. So they have this unique characteristic of differentiating into any type of cell type. So we can use this for regenerative medicine. For example, if they have any spinal cord injuries, for Parkinson's diseases, and so on. Induced pluripotent stem cells, they are reprogrammed adult cells, like I was talking about. So this type of stem cell can be used for patient-specific treatment. For example, you can create disease models, you can also test drugs, you can also try to have very specific treatment for a unique disease or for a person who requires a very special treatment. The other type of stem cells are hematopoietic stem cells, where they have the ability to differentiate into different types of blood cells. So such stem cells are used for blood-related disorders like leukemia, sickle cell anemia, etc. And we have neural stem cells, where they have the ability to differentiate into neurons and also other support cells. So this can be used for neurodegenerative diseases like Alzheimer's, or even for spinal cord injury. So there are a few disease-specific therapies that are being studied extensively. So for example, myocardial infarction, we have mesenchymal stem cells, which was discussed in the other presentation. We have diabetes, for which iPSC cells are studied extensively to make sure that insulin production is restored by these stem cells. We have autoimmune diseases like multiple sclerosis, where hematopoietic stem cells are extensively studied. Retinal degeneration diseases like age-related macular degeneration, where retinal pigment epithelial cells from stem cells are being studied to see if they are able to replace the RPE cells that are already in the body to restore vision for these patients. So stem cell donation and awareness. So I just wanted to talk a little bit about what type of awareness is there, and what type of agencies are there that help out with stem cell donation, etc. So one thing is that right now we do have a lot of talk about stem cell. There are a lot of people who know about stem cell or who know partially about stem cell. So it's very important that the messaging about stem cells and the stem cell therapy and the reality about how far the research is and how much in reality we can treat diseases with stem cell. So there is this one organization which we, the CERM students, had the opportunity to work for, is Be the Match. So there are non-profit organizations like Be the Match who advocate and who go around, have tables and go to events and try to get people to donate stem cells. So the registration process and everything is pretty simple. And so agencies like them, they try to educate people about stem cells. They try to educate people about how the process of donation is and how people actually require stem cell donation for treatments that are already available for them. So that's the good side of the awareness, right? And now we have the other side of the awareness, which is stem cell tourism. So this is one of the main issues when it comes to partial awareness about stem cells. So stem cell tourism is nothing but when people go to other countries seeking stem cell therapies that are not regulated or not approved in their home country. So for example, let's say that I need stem cell therapy, like I have some issue and I need stem cell therapy, and I know that this particular stem cell therapy is not approved in the US, but
I'm
it is
so sorry. Cypriya, sorry to interrupt. My apologies. But can we
Yeah.
wrap this up quickly? Because the other group will not get much time to present then.
Sure, Professor. So stem cell tourism is basically when people move to another country to get stem cell therapies. And this is an issue because there could be a lot of issues like health risks or legal issues where they cannot sue that particular hospital if they have any issues because of the treatment. So it is very important for us to know and to educate ourselves about stem cells before we move forward with therapies. So I would like to pass over to Reina for the next few slides.
Hello, I'm Reina, and I'll be talking about the stem cell transplant for leukemia treatment. So stem cell
Reina,
transplant.
we'll have to make it really quick, Reina, because the other group will only have 10 minutes to present. Try to wrap it up in like 1 minute or 2 minutes, please.
I don't mind staying later to make it a longer class.
Yeah, I'm fine, but I don't want to be inconveniencing the other OLI members. If you are okay staying like another 5 minutes, that's fine.
Nitika,
I'll try
that's
to
fine.
wrap this up
I
as
can
soon as
keep the meeting
possible.
open extra time. That's fine.
Sure. Thank you so much.
I'll try to be very quick. Okay, so as Sahithya mentioned earlier, stem cell transplants help with leukemia by enabling the body to replace damaged blood cells with healthy ones. So there are two types of stem cell treatments, which are autologous transplantation and allogeneic transplantation. Allogeneic transplantation is the most common type of stem cell transplantation, and it uses the stem cell from a donor, whereas autologous comes from the patient itself. Next slide, please. So, stem cells are collected from the donor or the patient, depending on the type of transplant taking place. In the past, physicians would extract bone marrow from a patient's hip bone in order to access the stem cells. However, today, most physicians use a process called pheresis to separate the stem cells from the bloodstream. So 90% of stem cell harvest for leukemia is now done with peripheral blood. And bone marrow transplant may still be needed when the patient's bone marrow has been destroyed by disease, or in cases where doses of chemo or radiation needed to cure the cancer are so high that a person's bone marrow stem cell will be permanently damaged or destroyed by the treatment. A stem cell treatment is a months-long process that can be physically and emotionally taxing, though the procedure itself does not take a long time. But the patients will have to go into the hospital at least 1 week before the transplant for a preparatory treatment that involves chemo and radiation, which is to kill as many cancer cells as possible before the transplant takes place. So after the transplantation, patients will remain in the hospital for recovery, and it can take 6 to 12 months or more for blood cell counts and the immune system to reach a normal level. While 80 to 90% of adults with acute lymphocytic leukemia will have complete remission, at some point during treatment, half will experience a relapse. And that'll be all. Thank you.
Thank you so much. You did a great job. Are there any questions? Maybe we can take one question at this point for our students. Okay, if not, you can always send us the question later. We will
I have a question, Nitika.
Yeah, sure.
does the what's associated, like the pain of getting the blood out of the bone marrow, stop people from wanting to be donating their bone marrow blood?
For sure. Actually, when we were advocating in Be the Match, there were a lot of people that expressed their concerns about bone marrow transformation. But right now, that's all day practice. But currently, all the procedures are completely painless. You'll be sedated, and it just takes like 15 to 20 minutes procedure, and you'll be discharged the same day. And even the blood collection, it's like normal blood donation, except that they'll be taking your blood out, just extracting the stem cells and putting back all the red blood cells back into your body. So both the process of stem cell donation is extremely pain-free and easy right now.
Thanks so much. That was great. But I just wanted to be giving enough time to group number 3, who have also worked really hard to present. So please, group number 3, kindly go ahead.
Yeah, I'll be sharing my screen. Can you see my screen?
Now we can.
Okay.
But make it bigger.
Can you guys see it now?
Yes.
Okay. Yes. Hi everyone. So it's just me and Kanwal. We'll be presenting on Sanfilippo syndrome. Kanwal?
Yeah. Am I audible?
Yes.
Hi everyone. My name is Ken. Hi everyone. My name is Ken. So today me and our team member Shruti will be presenting. For some reason, I'm hearing my echo.
No, we can hear you fine.
Okay.
Zio, lower your volume.
If you have
So
two devices,
today-
sometimes you get feedback.
Oops. Yep, there is an echo.
So Michael, what has to be turned off? Because it seems like two devices are doing the audio.
You might try turning off the audio, leave the microphone on, but turn off your audio. It's probably creating a feedback loop.
So Kamal, just follow what Michael just said.
So we will be presenting on Sanfilippo syndrome and the possible treatment using
Constant
stem
echo.
Yeah, there's an echo. I can hear my voice.
So Kamal, let's do one thing. Why don't you switch to this from her laptop, and you mute yourself. Let's try that.
Dr. Parmar, I have an iPad, so I'm not sure. So if I'm able to share my screen.
Are you not able to see my screen?
Yeah, I'm able to see your screen.
Okay.
Shruti, if
Shruti,
you
if you
can
can
mute
mute
yourself, maybe that would make a
Wait. Oops. I don't think I can mute.
Go ahead, Kanwal.
Okay, so what exactly is Sanfilippo syndrome? This Dr. Parmar, I'm not able to present.
Shruti, lower your volume.
Are you broadcasting from a cell phone?
Yeah, I'm broadcasting from my phone.
Yeah, you might want to volume all the way off. So the audio volume is the same as if you were listening to music. Turn that all the way off. Microphone should remain on.
Is that better?
Yes.
Yeah, go ahead and start. Let's see.
So this condition is also called mucopolysaccharides.
Professor is echoing.
So it's a condition in which mutations.
Shruti, can you just send the slide to one of us? We can present.
Okay, let me try. Okay, I just sent it. Let me know, Jemima, if you got it.
Yeah, just a second. Is the screen visible?
Yes.
Yes, thank you.
Go ahead.
Okay, so I'm so sorry for the delay, but my name is Canval. So today, me and our team member Shruti will be presenting on Sanfilippo syndrome and the possible treatments using stem cells. Can we go to the next slide? So what exactly is Sanfilippo syndrome? This condition is also known as mucopolysaccharide. It's basically a condition in which mutations occur in the lysosomal enzyme coding genes, which causes the problems in the stepwise breakdown of the aminoglycans, which are the basic for this hereditary disease. When one of these enzymes are lacking, the substrate cannot be broken down properly, which cause this particular disease. And these mucopolysaccharides are like huge complex polymers that are widely dispersed in most of the tissues, and they are composed of linear repeating sulfated acidity and amino sugar disaccharide units. Next slide, please. So based on their phenotypical and clinical characteristics, this condition can be divided into various types like MPS 1, MPS subtype 1, 2, and 7 for skeletal, brain and soft tissue storage, and so on. Next slide. Talking about the symptoms, children with Sanfilippo syndrome do not show very obvious symptom of the disease at the time of birth, but with age, storage of these glycosaminoglycans increases and it start affecting bones, skeletal tissues, and skeletal structure, brain, and all the different organs of the body. And for instance, image on the right side shows a very mild phenotype of the patient suffering from Sanfilippo syndrome. These are some of the symptoms that, and these are the symptoms that varies from person to person, like mental retardation, coarse facial features, skeletal irregularities, and so forth. Next slide, please. These are some of the symptoms that have been seen in patients, like challenging behavior, sleep disturbances, cardiac diseases, and so on. Next slide. So the Sanfilippo syndrome is comparatively a rare disorder, and there are a variety of factors in terms of geographical areas as well that plays a huge role in occurrence of the disease. In the US, it's like one child per 70,000 to one child per 160,000 births every year. And apart from that, if I'll talk about the situation worldwide, the stat revolve around one child per 50,000 to one child per 250,000 per year. Next slide. As we know, this condition is caused by a genetic deficiency or mutation that prevent the body to break down the heparin sulfate. Here are some of the examples. For instance, MPS subtype 3, the gene is involved SGSH, and this is the enzyme that had undergone mutation or genetic deficiency to cause this disease, and so on with all the other types that are listed here. Next slide. Now over to Shruti.
Yeah, I'll be talking about the pathophysiology. So in this one, the lysomes of like the genetic mutation will cause the enzymes to be deficient. And then the heparin sulfate will begin to build up in the lysozyme. Next slide, please. So some of the consequences of this is like the accumulations of the heparin sulfate will lead to serious consequences such as like neurodevelopment delays, neurological deteriorations, organ damage, and then growth problems, behavioral problems, etc. Sanfilippo syndrome, so like the ultimately leads to an early death wearing on the subtype. Patients with subtype A will have a life expectancy of up to like 19 years, and other subtypes will have life expectancy of up to like 30 years. Next slide, please. So some of the ongoing treatments there's currently no effective cure for Sanfilippo syndromes, but these treatments can assist on patients and prolong their quality of life. Next slide, please. So I'll talk about enzyme replacement therapy. So in this one, it's a specialized treatment approach targeted at individuals with this rare genetic disorders. So with this therapy, synthetic versions of this missing enzymes for each subtype, the missing enzymes are introduced into the patient's bloodstream through regular infusions. And these enzymes needs to be broken down, accumulated sugars that lead to a cellular and organ damage. So with this enzyme replacement therapy, it again, it doesn't fully cure Sanfilippo syndromes, but it will improve the quality of life, providing a much more manageable and supportive approach to managing the condition. Next slide, please. So for gene therapy, it introduces the functional copies of the mutated genes responsible for the disorder for each subtype into the patient's cells. So what it does is it typically is achieved by using vectors such as like modified viruses, and it's delivered to the corrected genetic materials of the cells. And the goal is to be able to enable the cells to produce the missing enzymes that are deficient in each of the subtypes of Sanfilippo syndrome. And by addressing, like the goal is by addressing the root cause of this disorder at a genetic level, these gene therapies aims again to provide a good quality of life and prolonging the lasting or for permanent solution or permanent damage to the body. And it's still a developing field, so gene therapy does hold promise for offering a transformative treatment for individuals. Next slide, please. Last is the substrate reduction therapy. This approach reduces the accumulation of the complex sugars that build up due to enzyme deficiencies. So unlike enzyme replacement therapy, substrate reduction therapy, it induces external enzymes. So instead of focusing on the inhibiting of the synthesis of the accumulating sugars, thereby decreasing their production. And this is achieved through administrations of drugs, and they interfere with the enzymes responsible for generating these sugars. And by reducing the substrate load, reduction therapy, it slows down the production of the disease. Next slide, please. And some of the challenges that individuals face are like physical characteristics, like mentioned before. Some of the facial features are thickened lips, large tongue, big eyes. They have excessive hair growth. They have a large head. Their height is significantly reduced. Their feet, they have wide feet, curled toes. Their nervous systems are also impacted. So they have a loss of language skills. So by the age of maybe 6 to maybe 10, they start losing their language and are often, they just can't talk. They lose their motor skills. Sometimes they may have dementia. They have increased seizures. Their gait is highly impacted, so hence their walking is also impacted. They have trouble sleeping. They have behavioral issues, tantrums, hyperactivity. They also have stomach symptoms, like they probably have frequent diarrhea. They have umbilical hernia. Some of their musculoskeletal symptoms, so like they might have arthritis, hip pain, carpal tunnel syndrome, and their eyes are also impacted, ear, nose, throat. And then their heart is also impacted. So sometimes they might have like really bad heart rates, high, low blood pressure. Next slide, please. And that's it. Thank you so much.
Thank you so much to all the three groups who worked hard on these presentations and did a great job. Are there any questions from the members for any of these three groups of students?
Okay, it seems like any of the things that you mentioned for this Sanfilippo syndrome could be considered in all different sorts of diseases or diagnoses. How do doctors even come to that diagnosis? And is the incident of the map you showed, where it's obviously more that in different parts of the country, is that because the diagnosis is more prevalent, is because the doctors are looking for it more there?
Can you please repeat the question?
Well, it seems as if that syndrome could go undiagnosed very easily. What would cause a doctor to start looking for that as a diagnosis?
I think, as Shruti said, gene therapy would be a possible treatment for this one. And there are a lot of ongoing research on gene therapy specifically.
All right, so if there's no more questions, I guess we can end here. And the OLLI members are encouraged to either email me directly if you have any questions, and I can always forward those to my students or try to answer them myself. So thank you so much, everyone, for your time. Sorry for going a little bit above time. It's almost 12:11 right now. But we thank you for your patience and your commitment and your excitement. So thank you very much until we meet again sometime soon with another breaking edge technology for discussion. Thank you.
Thank you all. That was fantastic.
Bye-bye.
Thank you so much.
Thank you. Bye.
Hello, this has been very interesting. I understand that about 60% of fertilized eggs are aborted, sometimes even before the woman knows she's pregnant. Is there any chance of getting some stem cells from those naturally aborted events?